Hi, in today's episode, we shall discuss about histology of different types of muscles. At first, to identify muscle, we have to look for long fiber-like structures lying parallel to each other. Here you can see long fiber-like structures present and they are almost parallel to each other. They are stained pink because they take the eosin stain, and they have a purple nuclei within which is stained by hematoxylin. You also know that the muscles are of three types, that is skeletal muscle, cardiac muscle and smooth muscle. While differentiating the muscle types, you have to look for three things. Firstly, whether striations are present or not. Secondly, whether branching is present or not. And finally, look carefully if you can find intercalated discs. Now, the smooth muscle are so called because they do not have that dark and light band patterns that you can see in the skeletal and the cardiac muscle. Now, these alternate patterns are called striations. So they are characteristic of skeletal and cardiac muscle and they are absent in smooth muscle. Hence, the smooth muscle is so named. Why are the striations present? Because these are due to the presence of actin and myosin proteins in the myofilaments. But that doesn't mean that the smooth muscles do not contract. They surely do. Just that the proteins are not this nicely organized in striated manner in the smooth muscle. Hence, the smooth muscle appears smooth. So now we come to skeletal and cardiac muscle where we find striation in both of them. So we have to differentiate these two based on some other features. What are these? We have to look for the shape of the nucleus, whether branching is present or not, whether you can see intercalated discs. Now, in skeletal muscle, the nuclei are flat, as you can see in this picture, often multiple and placed at the periphery of the muscle fiber just beneath the sarcolemma. So, what is sarcolemma? It is as the cell membrane of the muscle cell, and the muscle fibers are aligned in parallel fashion without any branching. In cardiac muscle, however, the nuclei are larger, oval or roundish and placed at the center of the cell. They are generally single in number. The fibers here are parallel, but they also show branching. The striations in both the cases, as you can see from the picture, they are in a direction which is perpendicular to the long axis of the fibers. In the same direction, we can see another feature which is specifically found in the cardiac muscle, and these are called intercalated discs. These are single strokes, discrete single strokes at intervals. I will show this in the later slides. Now coming back to the smooth muscle, here the smooth muscles are flowy, they appear in a flowy, wavy pattern and they have a flat nuclei within, generally a single nuclei at the center. Now we are coming to the slides which I have taken from Diffio's Atlas of Histology. In the first picture, you are seeing the skeletal muscle where you can see the stain used is hematoxylin and eosin. So there are striations, alternate dark and light bands, that is the A and the I bands due to the actin and myosin filaments. And you also see at the periphery there are flattened nuclei. Now both the longitudinal and the transverse sections of the muscle has been shown here. In the next slide, you are seeing this wavy long fibers with striations, but there are branching as well. So this is cardiac muscle. Not only that, you see that the nuclei are roundish, placed more or less at the center, and they're single in number for each muscle fiber. If you look closely, you will see distinct vertical strokes, darker than the normal striations, in the same direction as the striations, but a bit distinct and at intervals. These are the intercalated discs. This is a picture. The first one is of smooth muscle. Here you see the spindle-shaped cells with ovalish nuclei, and there are no striations here. So definitely it is smooth muscle. However, in the next slide you can see there are same wavy pink pattern, and that may be confused with the smooth muscle, but these are not smooth muscle. These are the collagen fibers. This is a specimen of dense regular connective tissue, which is often found in the tendons, ligaments, etc. And here you see that the collagen fibers, since they're non-living, they are not having any nuclei within. But the cells that you can see on the outer part, that is in between the collagen fibers, in the gaps between the collagen fibers, these are fibroblasts. They clump to each other end to end, numerous in number, and they are present in the gaps between the collagen fibers. Whereas if you see in the smooth muscle, in between the muscle fibers, the gaps are empty. Rather, the nuclei are present within the muscle fiber. That's the difference. That's how you will identify. But finally, you should also consider that which tissue you are looking at. For example, in alimentary canal, you will find the smooth muscle along the wall. So you will find in stomach, you will find the oblique muscle, the longitudinal muscle, this circular muscle, and rest of the alimentary tract you will find the longitudinal and the circular muscles. All these are smooth muscles. And connective tissue is also present. But in the alimentary canal, the connective tissue is not dense regular connective tissue. As I have mentioned, dense regular connective tissue is found in the tendons or ligaments. But in the alimentary canal, you may find, or rather you will find, this connective tissue in the lamina propria, in the submucosa, in the adventitia, all around. But this is generally this loose irregular connective tissue, where you do not find. And in this specimen, can you see side by side the dense irregular connective tissue and loose irregular connective? So these are irregular pattern. But on the other hand, in the upper picture, here you can see the flowy regular pattern of the dense connective tissue with the intermittent lines of fibroblasts. Here the fibers and the matrix and the cells, they're all haphazardly placed in the loose irregular connective tissue, which we also find in the walls of the alimentary canal. So that's for today. I hope you have understood how to differentiate between the different types of muscles under the microscope, and also you have understood how to differentiate between the smooth muscle and dense regular connective tissue, which you can often confuse.